Have you ever wondered about the ways in which humans have impacted wildlife communities? It has been well established that habitat loss, fragmentation, and degradation have drastically altered the ways in which species interact with their environments. As a matter of fact, the largest contributor to the extinction of species worldwide is habitat loss through urban and agricultural development. To put it plainly, if a species is unable to adapt to the rapid changes in their environment, they will cease to exist. Interestingly, in 2022, a study was conducted in Puerto Rico to observe the ways in which urban environments influence the genes expressed in a species of lizard called the crested anole. Genes are expressed in observable characteristics or traits that are referred to as phenotypes. Phenotypes vary within species and can also be influenced by environmental factors. Examples of phenotypes include an animal's coloration, their markings, and their size. Certain phenotypes can provide advantages or disadvantages that help or hinder an individual animal's ability to survive and reproduce. These beneficial phenotypes are often selected for or preferred by members of a species. Over time, environmental pressures can influence the phenotypic variance in a population of animals. For example, a tan-colored mouse would be camouflaged better on a sandy beach than they would be in a forest, and a dark brown-colored mouse would likely have a better camouflage for the forest. Because the tan mouse on the beach would be better able to hide from predators, the tan coloration phenotype and its underlying genes would be selected for in that population. In this case, the pressures elicited by predators influence the genes in the population of mice. This is the basis of evolution by natural selection. The crested anole study aimed to understand the ways an urban ecology influences the evolutionary dynamics of the species. Researchers observed populations of anoles in 3 distinct urban locations and the forests that were directly adjacent to them. They sequenced the genetic material found in the DNA of lizards in each population, and they discovered that there were 33 genes selected for in each urban anole population, regardless of how distant the urban locations were from one another, and more importantly, how distantly related the lizard populations were from each other. The lizards found in these urban settings had longer limbs and larger toe pads than their forest-dwelling counterparts. These differences in physical characteristics are speculated on being an adaptation to better travel on urban landscapes. This means urban environments can influence the evolutionary trajectories of species in similar ways. This phenomenon is called genotypic or phenotypic parallelism. These findings help us better understand the ways in which urbanization can influence evolution. They also help us think more broadly about the ways in which similar types of environments can place selective pressures on organisms that lead to phenotypic parallelism. Now this begs the question, what other animals exhibit phenotypic parallelism in urban areas? What about the western fence lizard that is so commonly found throughout Southern California? Have urban western fence lizards adapted to urban locations in similar ways to the anoles? Will western fence lizards found in the hills of the Cleveland National Forest have the same genetic makeup as those found in the adjacent cities? What about those found in the desert? What are the characteristics of species that are best able to adapt to this novel world? In our ever-evolving world, it's important to consider the ways in which we influence the environment and those that inhabit it. Thankfully, anoles were able to persist, but not every species is so lucky to adapt and thrive. Thank you for watching.